Good morning, my beautiful students. You're welcome yet again to your beautiful biology class. Today, we are going to be treating the topic, the biology of heredity 2, and the subtopic is going to be on exercises on gene transmission patterns. Exercises on gene transmission patterns. In other words, I chose this subtopic in order for us to practicalize most of the things we've been studying with some word problems that we're going to be solving. We have to solve some word problems just like you face in your external exams like WAEC, JAMB, NECO, and even some international exams, where they will give you some problems in genetics to solve concerning transmission patterns of genes, both in human and other organisms. So you're welcome to this class. Our specific objective at the end of this lesson, I should be able to define patterns of transmission of genes, state some major patterns of transmission of genes, and the last one, state methods of solving word problems in patterns of gene transmission. Let's define some terms that are important for us in this topic. The pattern of transmission of gene is defined as the way or trend in which a gene is transferred from generation to generation. The various patterns and changes that occur when a gene or group of genes are being transferred from parent to offspring. And it can come in diverse ways. For instance, a gene that has never been noticed in a particular family lineage, all of a sudden, you start seeing that gene appearing. It is a pattern of transmission of gene. It's taking a particular pattern. For instance, the gene might have been recessive all the while and has been in combination with dominant contrasting genes, and that is why it might not have shown in all the generations of that family until now. Or it might be as a result of mutation which has occurred along the line of the transfer of these genes from one generation to another. It could be of so many reasons. So it's also a pattern. You could also have patterns where you have complete dominance, where a particular gene outweighs the other one in its power of expression. And you can only have the recessive one coming out when they are in homozygous form. It's also another pattern. You also have another pattern in which a gene or two pairs of genes controlling a trait might be expressing themselves at equal strength. In such a case, you can call them incomplete dominance, or you call it what in genetics is called blending. So there are different patterns of transmission of genes. Even also in the sex-linked characters that we treated last time, it also had a particular pattern, and we studied that in detail in our last class. So now we're going to be doing some word problems, and we're going to be solving them. Now let's continue with our definition. It says the various modes of gene transmission reveals the behavior of gene in relation to other genes as they are being transferred and expressed from parents to offspring. So the next we are going to see is some of those patterns of transmission of genes. What are those patterns? Now we have the major one. There are so many patterns. In our modern scientific researches that are going on, there are different patterns that are being discovered day after day. But we are going to look at these few major ones. Number one pattern of transmission of gene is what is known as the pure stock transmission or the true breed pattern. The other one is the complete dominance pattern, which can produce a hybrid. The last one we are going to look at is the incomplete dominance transmission pattern, which can also produce hybrid offspring. Now, the above mentioned gene transmission patterns can be illustrated using the monohybrid crossing or dihybrid crossing methods, depending on whether it is a single factor inheritance or a double factor inheritance respectively. So what we are actually trying to portray in this last statement is that you can use monohybrid crossing to solve the word problem in order for you to understand the pattern in which the gene has been transmitted, or you use the dihybrid depending on the level of question asked. So let's look in detail concerning these different levels of gene transmission. Now, that first one we saw was pure stock. What do we mean by pure stock gene transmission? When a group of organisms belonging to the same species are crossed with the resultant trait being repeatedly produced from generation to generation, they are said to be of a pure stock or true breeds. You can see this very well in animals, all right? And also in humans in some cases. In animals, people that engage in farm animals, a lot of the time they will tell you that they don't want to crossbreed a particular pure breed they have of a particular animal with another breed from another country or from another region of the world. Why? They don't want it to be altered. They don't want their pure breed to be altered, maybe due to the quality of yield, agricultural yield they get from that particular pure stock. They don't want to interbreed it with another species. For instance, between lions and tiger, you can interbreed the two, and you get in between what is called a liger. Although I will not go into details concerning the liger. But then, the liger is a resultant crossbreeding between a lion and a tiger, although it cannot continue as a species for some reasons. But that is a hybrid as well. So, most people like to maintain pure stock, where a particular genotype is being repeatedly produced from generation to generation without any alteration along the way, either by way of mutation or any other means of producing a hybrid. No, is not allowed in it. If it continues to produce the same genotype, pure stock, from generation to generation, you can call it a true breed, or you call it a pure stock. An example can be seen when red flowered plants continue to breed red flowered offspring from generation to generation without producing another colored flower. This is possible. You can get a particular plant where the flowers are red in color, and from generation to generation, each time you breed them among themselves, they will continue to produce red flowered plants. From generation, first filial generation, second filial generation, and the rest of them, they will continue to produce red. Then you can call them the pure stock red flowered plants, or you call them the true breeds. This is one pattern, and let's solve a problem in this regard. As we can see in the next slide, the word problem is what will be the genotypes of the second filial generation of a true breeding red flowered hibiscus plant when they are crossed. What will be the genotypes of the second filial generation of a true breeding red flowered hibiscus plants? Now, I'm going to be showing you on your screen this particular method, how it works. Now, as you can see, the question said what will be the genotype of the second filial generation, which means since they are pure stock, the parent generation is going to have, for instance, something of this nature. They will have red color, whether it is in the dominant level or in the recessive level, they are going to be produced. Genotypes will be the same. So they are going to be homozygous in their genotypes, and that is a pure stock. So if you crossbreed it with another hibiscus that is pure stock, in their segregation of genes, they will produce red genes in their gametes. No matter how many times you segregate them, they will produce. Therefore, if you make their first generation, what you're going to get is all of them will be red flowered hibiscus plants. This is pure stock. There is no intrusion with any other type of gene coming into it. So it's a pure stock. Now this first one is the F1 generation, the first filial generation. But the question asked us, what will become of the second filial generation? Is there going to be any alteration? Well, I don't think there will be. Since the first filial generation are still having the homozygous dominant red genes, in their offspring of second generation, they will still produce the same stuff. In their segregation, they will produce the red and the red here, and when it comes down to their generation, they will all still be red colored plants. Their flowers will still be red. So as you can see on your screen, throughout the generations, they will continually be producing red colored flowered plants. And this is an example of a pure stock breeding or true breeding trait. Now, the next thing we are going to look at will be this next one, the complete dominance pattern. The complete dominance pattern. Yes, we have treated this in our previous topics. Let's treat a word problem with it. What are the genotypic and phenotypic ratios of the first filial generation of a cross between a true breeding white flowered rose plant and another true breeding red flowered rose plant, if the parent gametes are in the following genotypes? White flowered rose plant has homozygous recessive white genes. Homozygous recessive white genes, as you can see on your screen. While the red flowered rose plant is having homozygous dominant red flowered plants. Now, going by these genotypes of the parent gametes, you can easily find out and solve the problem when it said the genotypic, find the genotypic and phenotypic ratios of the first filial generation when these two individuals are crossed. Let's look at the result in the next slide. As you can see in your next slide, we have a red flowered homozygous dominant genotype and a white flowered homozygous recessive white flowered plant. When you crossbreed the two, you're going to get, in their genotypic ratio, you're going to get 1:2:1, the ratio. One homozygous red plant, and as you can see on your screen, you're going to get those following ratios: one homozygous dominant red flower plant and two heterozygous dominant red flower plant. Then you're going to have one homozygous recessive white flowered plant. Well, that is for the second filial generation. That is for the second filial generation. But the first filial generation, and that is the one down here, that is the genotypic ratio I've called. But the question asked us for the first filial generation. For the first filial generation, what you're going to get, all the offspring of that first filial generation are going to be red, and these are the first filial generation. If we are going to do the crossing, you will see that all the offspring of the first filial generation will be red. But in the second filial generation, you're going to get 3 that are red and 1 that is white. But let's work it out and see if the first filial generation, as is shown here, whether it is correct. Now remember that the red flower plant is dominant and is homozygous dominant, while the white flower plant is also dominant and it is recessive white. Now, if you begin to cross the two, you find out that you're going to have, among the first offspring, you're going to have this kind of genotype. Now, the next one, you're going to have this kind of genotype. The next, you will also have this kind of genotype. Then, the last year, you will also have this kind of genotype. So when you do the crosses, you're going to find out that the first filial generation, all of them are going to be red, but their genotypes are also the same. They will be heterozygous dominant red flowered plants. Phenotypically, all of them will be red, and genotypically, they are heterozygous dominant red, with a recessive gene to accompany them, which is the Y gene. So phenotypically, when you look at these flowers from the outward appearance, all of them will appear red, while the white color will be recessive, hidden. You will not see it. So the genotypic ratio for this one is 1:1:1:1, which is equal to complete one. The probability that the children, the offspring, are going to be red in color, the probability is 1. All right? Now, if you have to ask for the phenotypic ratio, it is also the same thing as the genotypic. The phenotypic ratio is that all the offspring are going to be red colored. So the probability that they are going to have red colored offspring is sure. Even if they have 100 offspring, all of them must be red. And so this is what happens when you have complete dominance in a particular region. All right? If you're crossing two of them that are of pure stock. So we have it as the answer for that one, just as it was asked. So the first filial generation, we have solved for that one. Then when we go to the next pattern of transmission of gene, which is known as incomplete dominance transmission pattern. In this particular one, let's look at a problem and solve it. What will be the phenotype of the F1 generation of a cross between two true breeding fowls of equal expressing color traits of white and black, with these genotypes that are already on your screen, where you have the white homozygous dominant white genes and also homozygous dominant black genes from the other fowl. So we have two fowls now that possess dominant, homozygous dominant genes. What will happen when you crossbreed two of them? What is going to be the resultant effect? In this particular case, we have the white color and the black color. What is it going to yield? Let's look at the next slide. In this particular slide, you will see a black fowl and then that of the white fowl. And according to the question, it says that two of them are having equal power of expression. Now, what will become of the offspring? In the offspring, when you mix black color, naturally in your ACT class, when they teach you about mixture of colors, you find out that when you mix black with white, automatically you're going to get gray color. So in this case, and these things are scientific researches that have been proven, it has been tested and proven, and it worked. So when you have two dominant genes that are equally expressing themselves, in the case of these fowls, you have the black and the white mixed together, the offspring, resultant offspring, all of them are going to be gray in color, as you can see on your screen. All of them will appear gray color. That is the result of what is called incomplete dominance. When you have genotypes that are of equal expression. All right? Now, that is one case of incomplete dominance. Another case I want to talk about in incomplete dominance, which happens in human blood, is what is known as the ABO blood group. In the human beings, the ABO blood group is also showing a pattern of incomplete dominance, as we are going to see in this word problem. It reads, what is the phenotype of the offspring of a couple with blood group genotypes AA and BB, respectively? Now, let's quickly go to our board and find out what will become of them once they are crossbred, or once they, come together to produce offspring. In such a case, you have the question say that the first parent has what is known as the AA blood group, and then the second parent has the B blood group. Why did I write two of them in the two pairs of genes? Why did I write them in dominant conditions? Yes, the blood group A gene in human being is always dominant, and the blood group B is also always dominant. They have equal power of expression. So when you now produce offspring from these two couples, what you're going to be getting are offspring with AB blood group, because they are of equal power of expression. As you see, AB blood group, all the offspring are going to be AB. Why? Because this is showing incomplete dominance, or what is known as blending, as you can see on your screen. So the ABO blood group also shows the pattern of incomplete dominance, but this is only reserved for blood group A and blood group B. When it comes to blood group O is always recessive. So if it appears as a contrasting pair to any of these dominant ones, the dominant ones will still overshadow the blood group O. You can try it on your own and work it out and see what it will produce. Now, when somebody is of blood group O, it means that his genotype, there is no A, there is no blood group B, neither is there any blood group A. All you have in his genotype is OO. That is homozygous recessive O genes. And so that's when you have somebody that is blood group O. But here it is not the same. So this is another example of incomplete dominance, as you can see on your screen. Now, we are going back to finish up on the topic and solving of word problems. We are going back to finish up on that particular one. So, having solved for incomplete dominance, another example, I want you to do this following assignment, which I would have wanted to interact with you on the screen, but due to want of time, I want you to do it as an assignment on your own, and I know you can do it. Remember, we did it in our previous class. And what is the assignment? If a man who is colorblind marries a woman who is a carrier of color blindness, what will be the genotypic ratios of the offspring? That is number one question there. Then secondly, how many of the offspring will be non-carriers? In other words, you can say normal. Children that are normal, they are not carriers, they are not sufferers. How many will be so? Then the next one, how many of the offspring will be carriers? How many will be carriers? And then the last, how many will be sufferers? Now do this assignment and then submit in the Google Classroom. Thank you for watching and stay safe. Keep on reading. For what you do daily determines what you become in the future. God bless you.